Today, we are gonna talk a little bit about the DNA regulation. So, let's start. Let's start it with an idea. Like, if we look at a human genome, that not all of the genes are transcribed and translated at the same time. It do, it could depend on type of cell or depend on the environment for organism. If we look at a if we look at an unicellular organism like bacterium, where its bacterial chromosome might have a gene involved in digestion for a certain type of food. Say if that food is present and maybe its bacterium is related to some type of stress mechanism, like maybe it is under a stress, it does have to express its gene. If you have a gene that is a sequence of DNA that is a part of broader chromosome and RNA polymerase need to attach some place, and the place where RNA polymerase will gonna attach is the promoter, and then our and then RNA polymerase will transcribe the gene. If we talk about the prokaryotic cells, multiple genes group together that have a one promoter. So this promoter here is called a regulatory DNA sequence. Promoter is a sequence or a regulatory DNA sequence, and RNA polymerase will attach to begin transcription. All of these genes as a bundle. And if we have a promoter with multiple genes, that combination of promoter and the gene is that a combination which is also called an operon. Something in our environment might allow repressors to take actions, and repressor which is attaching to sequence of DNA after the promoter, and so it blocks the RNA polymerase from being able to transcribe. The repressor protein is repressing the transcription and regulatory protein sequence where it attaches to operator. So promoter is a regulatory sequence where RNA polymerase will attach, and again operator is a regulatory sequence where repressor can attach and keep that RNA polymerase from being able to perform a transcription. We might have a feedback mechanism where this protein can only do its job if it has if it has the other molecules that attaches to it, such as co-repressor, a tryptophan, and which is and also called as amino acid, can act as a co-repressor. The operon is for production of the tryptophan or amino acid and are essential for creating the polypeptides. These genes actually codes for enzyme that are used in making tryptophan, so trip E, D, C, B, and A. All are once they are transcribed into messenger RNA and translated into ribosomes, these enzymes are used to create tryptophan. If we are in a low if we are in a low tryptophan environment, the RNA transcription process, the RNA polymerase can attach to promoter and begin the transcription process. Transcription of these 5 genes into messenger RNA, which can be translated into enzymes. We have a direct inhibition. We can also have a direct inhibition where tryptophan can bind or interact with enzyme 1 so that it could not no longer complete to precursor. For prokaryotic cells, regulatory proteins are negative and therefore they turn off. But when prokaryotic regulation affects the structure of RNA polymerase, which turns on the large group of genes. So, A regulatory repressor protein is normally bound to operator, which prevents the transcription of the gene on operon. If an inducer molecule is present, it binds to repressor and changes its conformation so that it is unable to bind to operator and allows the expression of the operon. Repressor protein are produced. Repressor protein are produced by a regulatory gene, but they are unable to bind to operator in their normal conformation. However, their molecules called co-repressor are bound to repressor protein, causing the conformation change to active site. The tripoperon involved in the synthesis of the tryptophan is negatively controlled by repressible operon.